So far class we've talked about cells, what cells are made of, what's inside cells. Now let's talk about how cells move stuff, how cells transport their stuff around. So the first thing we need to know is that all transport is controlled by the cell membrane. So right next to cellular transport, write the word cell membrane, circle it. The cell membrane is this phospholipid bilayer. It's got these fats and these phosphates attached to it, and it's a bilayer because it's got two parts. Now, when I say it is semi-permeable, all that means it is super selective in what it lets through. So it's going to sometimes let some things through, sometimes not so much. When we transport things, we're going to transport them in one of two ways, either with energy or without. So let's talk active and passive transport. So you see a blank triangle. We've done this a long time ago. So let's go ahead and remind ourselves. Here's how we determine whether it's active or passive transport. Let's pretend this triangle is a hill. Now, if I am going to walk up the hill, that's going to take some energy. And if it takes energy, then that means this is going to be active transport. The type of energy that active transport uses is ATP. Now, once I get to the top of the hill, I'm pretty golden. I can just slide right on down. It really doesn't take energy to do that. If it's something that doesn't take any energy at all, we call that passive transport. Otherwise known as diffusion. Remember, that one does not take any energy. Now, if we look at the concentrations of these gradients, if we start at a low concentration down here and we move up to a high concentration, that is going to take some energy. That is going to be our active transport. Whereas if we start at a high concentration and we move down, that's going to not take any energy. That is passive. Those molecules are just going to be let through. When we are letting molecules in and out, when it doesn't take any energy, again, that's going to be the passive transport, otherwise known as diffusion. Now, along with that are going to be a few definitions that we need to know. So, this slide right here has all of these definitions. On your page, you need to write the word, the vocabulary word, and its definition. So go ahead and pause your video for a minute or so and quickly jot down these vocabulary words and definitions. All right, you should have those down. You should have paused your video and had those down. So now we're going to go ahead and kind of look and analyze this diagram. If I look at this diagram, the first thing I notice is that there's not many molecules on this side, but then there's tons over here. So these molecules are really crowded. They're going to want to move over to the other side. But instead of letting the molecules through, this membrane is going to do something different. Instead, it's going to let the water through. So it's going to use the process of diffusion in order to let these molecules through. So we're going to label this one is diffusion. And if I go over here, notice how there's different water levels, but all of the molecules are separated at this point. They're happy. They have their space between them. That is when we have finally reached our balance. That is finally when we have reached homeostasis. Everything is equal. Everything is balanced. We are good to go. Now, all of this really is talking about the movement of water instead of molecules. When we are only moving water, that is when we call it osmosis. Now, the last thing we're going to talk about is how this actually looks like when we are visualizing a cell. So we did our egg experiment in class, and we put them in different solutions and saw that they changed shape and changed color. Now let's kind of look at maybe what a plant cell might look like when it is placed in different types of solution. The last thing that we're going to annotate on our homework notes for our review today is going to be what this looks like in a cell. So we have three types of solution. The types of solutions that we're looking at are going to be either hypertonic, isotonic, or hypotonic. So when I say the word hyper, my mouth gets smaller. In order to make something smaller, you're going to have to take something out of there. So that means the resulting cell is going to shrink. It's going to get smaller. Now notice how in isotonic we have one arrow going in and one arrow going out. That's the same, so it's going to be equal. There is going to be no change. That cell remains the same. Hypotonic is the opposite of hyper. Hypo is going to grow because water is being put into that cell, and that cell is going to get bigger and possibly explode.